So good day, everyone. I'm Curtis Rambaran, Senior Director and Physician at Daiichi-Sankyo, involved in the development programs for Duchenne muscular dystrophy. And today I would like to present an overview of Daiichi-Sankyo's Duchenne muscular dystrophy programs. So firstly, I will give a brief corporate overview of Daiichi-Sankyo, then go on to describe our leading asset in the clinic for Duchenne muscular dystrophy, DS5141B, a bit on the preclinical data, then the clinical program to date, and the next steps or future plans for the program. So Daiichi-Sankyo is a global pharmaceutical company. We have got a worldwide presence in more than 20 countries, with several manufacturing sites globally. And we have a very strong history in the development of cardiovascular therapies for cardiovascular diseases. And currently, our near and short-term future vision is to be a global pharma innovator with a competitive advantage in oncology and also in the development of therapies for the rare diseases. This is a snapshot showing our presence from east to Europe to west, so very global. And our headquarters in the US is based in Baskin Ridge in New Jersey. And currently, we have a number of approved therapies on the market in the US for hypertension, for thrombosis, dyslipidemia, and IV iron therapy, and very importantly, recently, therapies for tenosynovial giant cell tumors and metastatic breast cancer. So that gives a flavor of our approved therapies currently on the market. And here is a snapshot of our major research and development pipeline around specialty mixed medicines and vaccines. And you'll see that gives a spectrum of therapies for cardiovascular disorders, for pain, for the rare diseases, including Duchenne muscular dystrophy, pseudoxanthoma elasticum, and also for atopic dermatitis, and a number of novel vaccine programs. And in the red box, I've highlighted our primary program in Duchenne. It's a DS5141, which is a antisense oligonucleotide, and that's been given a Sagekaki designation in Japan. And that is really intended to designate drugs with prominent efficacy in the disorders that are serious, life-threatening, and to allow them to become first in Japan. And that gives a priority for clinical trial consultation and review. So here is the chemical structure for DS5141B. It's an antisense oligonucleotide against Exon 45. And it's very novel. It's a chimeric consisting of a combination between a 2-methyl RNA or second generation oligonucleotide and an ethylene bridge nucleic acid, which is very much a trademark of Daiichi-Sankyo. And in the ENA, the ethylene bridge nucleic acid, there is a strong bond between the oxygen atom on 2' of the ribose ring and the carbon atom of the 4' position. And this leads to a ribose ring that's an ideal conformation for the traditional binding, or what's known as the Watson-Crick binding, and confers special stability on the compound. So chimeric, that could be considered to be a third generation oligonucleotide. And this gives, from a chemistry perspective, some of the potential advantages. You see that the affinity towards messenger RNA is very high, and the nuclease resistance also is very high as you compare across the first and second generation oligonucleotides. So this gives a lot of confidence very early with regards to the novel chemistry. So the mechanism of action, here's an example of exon 44 deletion, which is a mutation that's amenable to exon 45 skipping. So traditionally, with an exon 44 deletion, as you translate from the pre-messenger RNA and activate the splicing mechanism, you would lead to an out-of-frame mutation. So you don't have very good lining of the exons, and that will result in a non-functional dystrophy, typically like you see in Duchenne. With antisense oligonucleotide like DS5141 that's targeting exon 45, you're masking exon 45, which is very next to the mutated exon 44 or the deletion. And this interferes with the splicing mechanism and then leads now to an kind of masking 45 to an in-frame mutation. And this will result in a partially functional dystrophy, and very much like you see in the Becker-type muscular dystrophy. So this is well known. And very much the mechanism of action of DS5141. Here is some preclinical data. So this shows data from the MDX mice after weekly administration for 4 weeks of DS5141B, a PMO ASO, and a 2-O-methyl ASO at various doses. And the objective here was to look at the degree of exon skipping in the different beds, the anterior tibial muscle, the diaphragm, and the heart. And you would see that in the vertical deep red bars, there is significant exon 45 skipping observed in the anterior tibial muscle, the diaphragm, and in the heart, particularly and predominantly for DS5141B. And that was at over 3 milligrams per kilogram. So this, together with the basal chemistry, gave confidence to then proceed into a first-in-human study with the potential for likely differentiation versus other earlier therapies. So the phase 1-2 study, the first-in-human study for DS5141B, was designed to assess the safety, the tolerability, the efficacy, and the pharmacokinetics of DS5141B in patients with Duchenne muscular dystrophy. It's multi-center, open label, and very importantly, this was delivered subcutaneously once weekly. The study was in Japan, 2 principal centers at the National Center of Neurology and Psychiatry, Dr. Komaki and Dr. Owano at the Kobe University Graduate School of Medicine. The inclusion criteria was typical for some of these studies, ages 5 to 10, ambulatory patients with at least 325 meters on a 6-minute walk test, mutations amenable to exon 45 skipping, and patients who were glucocorticoid naive or had been on a stable dose of glucocorticoids. For the exon exclusion criteria, really to highlight that patients with impaired ejection fraction would be excluded. Here is the study design. So the study was in 2 parts. Part 1 was the dose escalation for first-in-human. You had 2 cohorts, each with 3 subjects. Each cohort started at 0.1 milligram per kilogram for cohort 1, cohort 2, 0.5 milligram per kilogram. And you had a DS5141B subcutaneously administered once weekly for 2 weeks, so 2 doses. Once that was safe and well tolerated, you then escalated to 2 milligrams and 6 milligrams in the same patients, again, for once weekly administration for 2 weeks, 2 doses. At the end of part 1, the safety was deemed sufficient and tolerability to proceed to part 2, which was the same subjects again, administered 5141B subcutaneously once weekly for 12 weeks at the 2 dose levels. The yellow stars reverse to the muscle biopsies done at baseline and at the end of part 2, and this was in the thigh muscles. And note also that we had an additional subject that was recruited at the end of part 1, so we ended up with 7 subjects dosed in part 2 of the study subcutaneously once weekly for 12 weeks. Here are some baseline characteristics. So the patients range from ages 5 to 10, exons amenable to 45 skipping, 6-minute walk ranged from the lower limit to 325 to 422 meters. And there was normal left ventricular ejection fraction at baseline. And all but 2 of the subjects were currently taking steroids. Reassuringly, there were no serious adverse events. There was no adverse events that led to discontinuation of the study drug, and all adverse events were considered to be mild. Now, here is a summary of the adverse events that was experienced in at least 2 patients. So, of significance, you had see the injection site reactions, and you had in part 2, 4 subjects, 2 at the 6 milligram dose, 1 and each one at the 0.5 and 2, and in part 2, 6 milligrams, again higher at the higher doses. But very importantly, these injection site reactions were mild, as reported by the investigator and reviewed, and they recovered before the next dose. And so far in the ongoing study, which I'll speak about a bit in a short while, we've seen any subcutaneous side reactions that resulted in discontinuation of therapy or serious unwanted effects. There was also noted some changes of microglobulin in the urine, predominantly in part 2 of the study. In terms of efficacy, we looked at exon 45 skipping by mRNA expression and the protein expression in the skeletal muscles. So here is the exon skipping by RT-PCR, and you would see that in all of the patients, all 7 patients, exon skipping was observed, and this was confirmed by sequencing, and this data has been presented a year and a half ago at several meetings. So very reassuring for the mechanism, the pharmacology. I summarize here the data. So DS5141 was found to be safe and well tolerated after 12 weeks of treatment at both the 2 milligrams and the 6 milligram per kilogram doses. Exon skipping in messenger RNA was confirmed in all 7 subjects. When we looked at dystrophin protein production at the end of the 12 week, it was only observed in 1 patient. So therefore, a decision was made to extend the study to part 2 extension to examine DS5141B over a longer period for 48 weeks. That study is currently ongoing, and that's deemed phase 1/2 part 2 extension study. So the next steps would be to complete the part 2 extension of the phase 1, phase 2 study. That's ongoing. The top-line results is anticipated for December 2020, so later this year. Planning for success. If those results are positive, safety and tolerability proven, and efficacy, we'll proceed to develop a US global program for starting in 2021. We also have a number of follow-on programs using the novel ethylene-bridge nucleic acid technology that I showed in the early part of the presentation. And this is targeting multiple other exons in Duchenne. So given an overview of the program so far, and this would not have been possible without the kind support and participation of the Duchenne patients and families in Japan who are kindly participating in the trial, and even through the current COVID crisis. This has interestingly not affected participation in the trial so far, which is really, talks to the dedication of the patient and investigators. We also are grateful to Drs. Matsu and Takashima, who was integrally involved in the ethylene-bridge nucleic acid chemistry and development, the Orphan Disease Treatment Institute in Japan, and of the project team from Japan, who was kindly agreed to share vital information, and our specialty medicine clinical development team in in the US, which I'm representing. So thank you very much for your time, and we'll certainly provide more details and events as they get released during the study. Thank you.